Hi, everyone. Let's get started. Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Please type any scientific questions in the Zoom Q&A box. All attendees can prioritize the questions by clicking on the thumbs up like button. Questions will be responded to at the end of the presentation. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions will also be provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, Dr. Jose Conejo Garcia. Dr. Jose Conejo Garcia is the chair of the Department of Immunology and co-leader of the immuno-oncology program at HLA Moffitt Cancer Research Institute. Jose, take it away.
Thank you very much, Alan. And I am honored to introduce today Dr. Catherine Wu. She's the chief of the Division of Stem Cell Transplantation and Cell Therapies at Dana-Farber. She's a world-class investigator, a member of the National Academy of Medicine, a member of the Association of American Physicians, and she has a very awesome career. She graduated as an MD from Stanford and completed her clinical training in Boston at Brigham and Women's Hospital and Dana-Farber, where she has worked since she became an independent researcher. Her laboratory has focused on using genomic-based approaches to discover antigenic targets and to understand the molecular basis of the therapeutic response as well as resistance. Recently, she has led early phase clinical trials to test personalized tumor vaccines in the context of melanoma and glioblastoma. So she basically has brought back vaccines and made them cool again. And I am, for one, very interested in her latest research and what she has to tell us. Dr. Wu, the floor is yours. Thank you.
Thank you so much for that very comprehensive introduction. Let me share my screen. And let's see. Here we go. Okay, so we'll get started. So yes, thank you so much for the introduction. It's really a pleasure to be able to speak to this audience. I thought I'd kind of give a little bit of background of kind of my scientific journey and kind of the things that where I've come from, but also what are the things that continue to excite me as we're in our current very exciting time and age. These are my disclosures, and I think this is the starting point. I think, you know, for all of us, we've always been hopeful that we could harness the specificity and the potency of human tumor immunity, the immune response in the service of eradicating cancer. But certainly it's been a very, very long journey. And I think that as we start to think about cancer and the immune system, we realize that it's really quite a complex and intertwined interaction. That we know ever more about tumor heterogeneity and clonal evolution and the ability for cancers to adapt. So even as there is an effective immune response, the cancer is continually evolving in different ways to inactivate this response, either directly or indirectly. And so a very difficult problem, but a problem that my lab has really very much been interested in embracing. And this is just a photo that we took on the at the invitation of the 25th anniversary of the journal Immunity, where different immunology labs were invited to submit a photo. And so this was a kind of a quick impromptu photo right after a lab meeting. And I think you can see clearly that there's different lab members who are working across diverse malignancies, and another part of the lab that is really thinking about how to harness immunity so that we can actually fight cancer. So I'll start with graft versus leukemia. So some of you may know that my clinical roots are in stem cell transplant, and I think in the current day and age, it's hard to think of a time where tumor immunity was not a part and a pillar of cancer care. But certainly, I was very excited about the prospect of curative therapy that could be achieved with immune responses, and so. Transplant, as you know, is an old therapy. It's been around for a long time. I want to remind our audience that it's been about 30 years since the Nobel Prize went to Donald Thomas for his groundbreaking work in establishing a stem cell transplant as a therapy for malignancies. And certainly it's currently part of the established therapeutic landscape for blood malignancies. You kind of see this kind of empty area, and then again so much new activity, and I think that serves to just emphasize that even as this is a very, very complex therapy, and so again I think this audience knows that stem cell transplant involves harnessing stem cells from a donor to give to a patient, and that stem cell product has the whole range of immune cells within the donor inoculum, and that there is engraftment and immune reconstitution that happens that serves to hopefully eradicate the leukemia. But I think what I want to point out is that transplant, although it is a old therapy and an established therapy, so many of the principles that we learned through transplant over the years have provided the foundation for all these new areas in modern day immunotherapy, topics that we're thinking about all the time. So what are the properties of the T cells that are actually the basis of the responses that we see, whether it's as TIL therapy or gene modified CAR T cells? What are the antigen targets that we're going after in transplant? I'll come back to this. We're always thinking about minor histocompatibility antigens, but you'll see as I talk that this is a natural bridge to thinking about neoantigens and personalized therapy. I think we worry about immune overactivation, and graft versus host disease is always the yang side of, or the yin side, whatever you want to say, the toxicities that we don't want to think about. And these days, as we think about cytokine release syndrome and autoimmune toxicities, they resonate very deeply with our ongoing struggles with graft versus host disease. And finally, the concept of combination therapy. I really consider transplant the ultimate combination therapy, where you have harnessing of cellular humoral immunity, you have cytotoxic reduction of added together with immunotherapy. So I think again that has great resonance with a lot of the different active items that we're thinking about these days. And so I think, as I kind of entered my training, I was very interested in allogeneic stem cell transplant, and specifically this setting of donor lymphocyte infusion for chronic myelogenous leukemia, because it was already known that 75 to 80% of patients who received lymphocytes, so in the absence of any further chemo or radiation therapy after stem cell transplant, that there was a sizable proportion of patients that would go into durable remission. So this, to me, at the time where there was really not other such robust examples of human tumor immunity in action, opened up questions of trying to think about how the different arms of immunity are coordinated, and opened opportunities for thinking about antigen discovery. So I had fun kind of pulling this one out. This was actually one of the first papers that I wrote as a postdoctoral fellow, and this was back in the day when TCR sequencing was actually done in these 100 microliter PCR tubes with mineral oil, and 26, 28 tubes per repertoire. So I think we've come a long way. But I think the point of this is to say that compared to a normal donor, already through this type of spectrotyping approach, we could see that prior to therapy, there are defects in the T cell repertoire that are very visible, using this type of assay. And what we could see is that in a patient that successfully reconstituted after 1 year, you can see that the peripheral T cell repertoire actually approaches what you see in a normal donor. And so I think that actually sets up this kind of concept that one of the advantages of transplant is thinking about how we can actually provide a competent donor immune platform from which we could then add in combination all the other goodies, such as vaccine-associated therapies or ways to enhance immune reconstitution, either by adoptive cellular therapy or cytokine therapy or whatnot. So again, I'll come back to this again. Just to close out this portion of the talk, I just wanted to share some of the data that we generated thinking about transplant in the, I would say, in the earlier era. And this was a study that I performed together with Pavan Bhattacharya when he was a resident, actually. And this was going back and looking at patients who had had a DLI response, going through the Brigham and finding all these marrow blocks and trying to score patient samples for T cell infiltration. And I think what we found was pretty interesting, which is that patients who were destined to respond versus non-responders already had higher T cell infiltrate even prior to infusion that thereafter expanded. And so I think this idea, we're always talking these days about kind of a T cell associations with ability to modulate response thereafter. And here we could see it already in the marrow happening with this kind of prototypical cellular therapy. I think also this was another early study that we did, which speaks to the ability to manipulate the immune response following reconstitution of donor immunity. And this was in the setting of patients with very advanced chronic lymphocytic leukemia who received a whole tumor cell vaccine admixed together with a cellular delivery approach for GM-CSF. So this was K562s that were modified to overexpress GM-CSF, and these were given in the early post-transplant period. And we were able to monitor a series of patients on this phase 1 study who had received 5 to 6 of these different personalized whole tumor cell vaccines, and we could test their peripheral blood for specificity against either tumor cells or different cellular components that could help us distinguish whether or not the responses were actually restricted to the tumor or related to minor histocompatibility antigens that were either restricted on hematopoietic cells or that would be broadly expressed. And so I think just to bring you to this plot, what we could show that was in fact these vaccine responses did in fact increase tumor reactivity as opposed to components associated with minor histocompatibility antigens. This was different than other comparison patients who had also gone through transplant, but who had demonstrated very high graft versus host disease. So there you see reactivity against all the different targets, as opposed to patients who received neither vaccine nor had graft versus host disease. I think we were able to take it one step further, and it turns out that underlying this response, at least in some patients, we could detect that there was response against predicted neoantigens, and we could even identify which neoantigen it was. And so shown here is, you can see that in this particular patient who had a long-term response after this type of whole tumor cell vaccine, that there was preferential recognition of the mutated versus wild-type neo-epitope, and that there was recognition of the patient's autologous tumor. And so I think this is kind of where things were before we heard that across the river that there was next generation sequencing that was coming along. And I think in that circumstance, it's really knowing that suddenly that there was an approach to scan the genome, open up the possibility of thinking about new types of antigen targets that could be targets of effective tumor immunity. And this is what brought us to neoantigens, because I think neoantigens is not a new concept. Neoantigens have been thought about for a long time as really the optimal tumor antigen, because These are antigens that are born of tumor mutations that we know that are part and parcel of tumors, and hence they have exquisite tumor-specific expression. They've always been anticipated to be highly immunogenic because they're born of somatic events, and hence there isn't central tolerance, and so like pathogens, highly immunogenic. And this is really different than where we had been in general as a field, thinking about shared overexpressed tumor self-antigens. I think for a transplanter who had already been thinking about minor histocompatibility antigens and knowing that single nucleotide variants certainly could cause clinically evident disease, such as graft versus host disease, it wasn't a big leap of faith to think that tumor neoantigens could also be immunogenic. And so when sequencing came along, we were eager to try to use this new genomic information and develop the pipelines that would allow us to systematically identify tumor neoantigens. And so just schematically, if we have RNA and DNA information from a tumor cell and DNA from a comparison match normal, and scan for the coding exome for mutations that are expressed, then certainly we can then also employ predictive algorithms based on the patient's own HLA typing and ask which of those mutations would bind to those personal HLA alleles, and then that would set us up for identifying candidate targets that, for example, we could use for vaccine manufacture. So I don't want to spend a whole lot of time on this because this has already been published and discussed at length, but certainly we perceived that neoantigens would actually really bring a paradigm shift and would allow us to think about how to launch immunity in a multi-pronged fashion that would be tumor specific and really open the doors toward personalized therapy. And so again, this is a conceptualization that we developed a while ago, but just thinking about if we could in fact procure tumor in normal, then could we not set up our pipeline so that we could predict peptides and then come up with a manufacturing strategy? At that time, we were thinking about synthetic peptides because of the evidence that had already been present to show that generation of personalized peptides would be feasible, that kind of pooling many different peptides together would not affect the patients adversely, and would provide us with a flexibility to really shake up the content of the vaccine in a real-time fashion. And so coming up with a synthesis strategy, but then also trying to think about how we could also combine together with immune checkpoint blockade inhibitors and really get this in a syringe that we could give to patients. And so we came up with NeoVax, which the goal was about 20 synthetic long peptides, building on the foundation of work by Case Malif and others that suggested that demonstrated that these long peptides, these 20 to 30 mers, could require a professional antigen presenting cell to process and present epitopes and hence coordinate CD8 and CD4 responses together. These were organized in pools and mixed together with best-in-class adjuvant that's widely available, poly-IC, LC, and that we would give as a sub-Q injection. And again, this is already published data, but I just want to point out that the first setting that we went after was in high-risk melanoma back in 2017. This was at the same time as the group of Ugur Sahin, and I'll talk about that a little bit, and following shortly after the work of Beatrice Carreno with Jerry Lynette using dendritic cell vaccines. And we've also ventured into newly diagnosed glioblastoma, also concurrently with a group in Germany. And in totality, these studies have shown this type of approach to be safe, feasible, highly immunogenic, and also anecdotally with promising results in combination with checkpoint. And so just to give you a flavor of the data, This was actually the results of immune monitoring of our first 6 patients. You can see across the bottom that the vaccines are given in a prime boost schedule, so priming just to get early antigen exposure and boosting then to encourage the development of immunologic memory. So mimicking what we hope is happening, for example, in a viral infection. And the different colors are the different pools. I mentioned the 20 different peptides, and we divided them into different pools to decrease immunologic competition. And I think what you can see very nicely is that actually in all of the patients we saw responses. And we saw responses against peptides in different pools, so many different colors, again, aligning with the goal of trying to generate responses across many different epitopes, not a single epitope, since we wanted to be mindful of the challenge of tumor heterogeneity. This is another kind of visualization of the type of responses. So having identified responses in pools, we also went through the hard work of trying to deconvolute which of the exact peptides within each pool that was actually responsible for the immune responses that we saw. We saw not only CD4 responses ex vivo, but also CD8 responses that required a single stimulation. And I think what you can discern through the red versus the black lines is that we, for the most part, saw really mutant epitope-specific responses as opposed to the analogous wild type. And across the 6 patients, we could see very nice CD4 and CD8 responses against the designed peptides. I think one of the most exciting results that we saw was in the example of these 2 patients out of the first 6. And these were both patients who had stage 4 disease. So after vaccination, it wasn't totally unexpected that both recurred after vaccine, but then their treating physician started them on anti-PD1 antibody. And what was remarkable is that after a mere 12 weeks, both of them went into complete clinical response. And I think you can see that, you can appreciate the melting of these lesions here after vaccine and then after 12 weeks of anti-PD1 therapy. What's been totally exciting is that having seen this, we asked ourselves, what happened in the intervening time? And we've been able to follow up with these patients even 4 years later, and by now they're off anti-PD1 and continue to be in complete clinical response. And in general, we started to ask ourselves, what do we see as we kind of take these patients out and look in the longer term? And so just in a nutshell, we recently reported out looking at these different features, namely, what were the transcriptional changes that we saw in those neoantigen specific T cells that were generated. We were able to sort out directly from patient samples, 3 different patients. We evaluated those neoantigen specific T cells directly from peripheral blood and perform single cell transcriptome sequencing. And what was actually seen in terms of those transcriptomes was exactly what one would hope with a vaccine, which is a nice cascade of responses going from naive to stimulation of cytotoxic and even immunologic contraction, and ultimately generation of memory that we could even see upon the boosting immunizations. We also saw T cell diversification and persistence. We could use TCR sequencing data to follow clonotypes over time. So in this example, where each color represents the first instance that we see a clonotype, I think what you can appreciate is that over time, you can see the red kind of persisting, but also the colors diversifying. So we see a great richness of the neoantigen specific T cells that are generated by vaccine. And then when we looked functionally, what was pretty exciting is that across all the patients that we looked at, shown on the first 3 columns are ELISpot data, where we looked directly after the first boost. So you can see strong responses to each of these immunizing epitopes, but then even close to 4 years later, we still see these circulating responses in the blood to the same exact immunogen. So that's actually pretty cool. We also in some instances, we were able to track those neoantigen-specific T cells to the site of tumor. So this is a little bit tough to see, but just if you can follow with me, this was one of the patients with stage 4 disease who had had a lung met, had gotten a vaccine, recurred and then got pembro. And we had already characterized, as I mentioned, the transcriptomes by blood. So you see here week 16 after the first boost, and then here in the middle of getting pembro after the vaccine. And then what the overlapping circles tell you is that really we see this was single cell TCR characterized, and we can see the same clonotypes for the most part, lots of overlap between the two time points, where in the red are the ones that we confirm are actually neoantigen specific. And I think what was super exciting is that layered on top of this, we were also able to characterize the bulk TCR sequencing of the recurrence sample. And looking at that, because we already knew at the single cell level which were the clonotypes associated with that were ascribed to neoantigen specific responses, we saw those same clonotypes actually present in the bulk sequencing of the tissue specimen, really supporting the idea that these same neoantigen specific T cells are also tracking to this site of recurrence. And one might hypothesize or speculate that it is this very tracking of neoantigens to the site of tumor that set the stage for such a robust response to PD-1 therapy that we saw following initiation of that therapy. Finally, a measure that folks are increasingly thinking about in terms of looking at on-target tumor responses. I want to remind you that this was a trial that was done in the adjuvant setting, so we didn't necessarily have tumor per se that we could follow over time to see whether or not there was a response to the vaccine. But certainly when we asked whether or not there was a secondary immune response suggesting that there was a release of epitopes following cytotoxicity of tumor, in fact, we did see epitope spreading. And this is an example of one patient where we see new responses even after vaccine priming and boosting, and then again upon anti-PD1 therapy. And this also mirrors what was seen in a trial run by Patrick Ott with Neon Therapeutics, where in a larger group of patients, epitope spreading was actually associated with better progression-free survival. Okay, so I think just some thoughts about our trial, which is, I wanted to mention that I don't think it was by chance that we reported out our melanoma neoantigen vaccine study at the same time as the group of Ugur Sahin. I think good ideas, science is incremental, and I think the time was ripe to think about how to use a genomics approach to design neoantigen vaccines in real time. I think what is remarkable, though, is that these are two different independent designs, independent groups looking at patients with melanoma, but really coming across designing studies with a different epitope length, numbers of epitopes, numbers of doses, but overall the immunologic responses that we're seeing across these two trials are relatively similar. And so I think that really speaks to the fact that there are probably many roads that lead to Rome. I want to reflect for a moment on what's currently happening with the SARS-CoV-2 vaccines, where globally we've seen all different types of delivery programs that have been generated by different companies in different countries, and they are all stimulating responses to more or less to a degree, one degree or another. Obviously, the group from Sahin was using RNA, whereas we were using peptides. And so I think it's really comes down to the bells and whistles that are used in terms of maximizing what these different delivery approaches can do. And again, I'm going to come back to this a little bit later. So I think now, as we kind of go forth, I want to kind of give a perspective in terms of where we are going next, because there are really, I think what we've achieved kind of with our proof of concept studies is simply that proof of concept, but really these studies raise so many questions, some of which, in order to make this more available, certainly there are improvements that we are already embarked on, but many, many more needed improvements that require much more effort. And so my group at large has been trying to address these different sort of topics, and I'll kind of run through some of them with you. So for example, can we do better at prediction? Because part and parcel to our process is taking the genomic data and trying to predict what are good epitopes to go after. And so for some time now, at the outset, we realized that the predictive algorithms were limited by the amount of training data. And so already some time ago, we generated an approach to actually generate the data that we would need to train these algorithms. And that was this idea of trying to take single allele cell lines, and rather than having the full complement of class 1 alleles, that we would try to control the situation, use these bold cell lines. So we use a B722, B721221 cells that are null for HLA, and then introduce a single allele, such that if you were to then pull down class 1 and elute off those peptides and run them through the mass spec, then there's unambiguous allele assignment, and then that sets you up for the stage to re-look at the rules of antigen processing and presentation. We actually took this further. So initially we looked at 16 different alleles, and then with some focus, we generated 95 different HLA lines in A, B, and C. And in doing so, we were able to collect 180,000 peptides. And this was really a very nice training set that would allow us to then generate allele and length specific models, as well as pan-allele, pan-length models. This data is publicly available, and I think has most certainly been already incorporated in some, for example, in NetMHC tools. But we certainly did create our own set of tools, which we call HLA Athena, which at the time that we reported this out, we were able to compare against other standard tools and show improved performance. And really where this actually comes into play is that certainly, I said before, I don't think it was by chance that a lot of these studies started in melanoma, because it's a unique profile of being high mutation rate and having lots of different sort of mutations accessible that one could then test and go after. I think as we start to go to more lower mutation burden tumors, where and you're trying to design, out of your 20 slots, how to make best use of the most immunogenic predicted peptides within those slots, I think improved prediction certainly makes a difference. Another question is, can we find new classes of targets by necessity? Our most robust predictions are for single nucleotide variants and insertion deletions, but what about RNA generated alterations? What about the products of fusion transcripts? And I just show here a recent paper that we reported out together with the group of Aviv Regev, where we were able to use RiboSeq, and by looking at the translation, it allowed us to consider unannotated open reading frames as but yet another source. As we go into a hostile environment, what do we know about that environment? Can we understand the cell states in natural disease and also in response? This was work performed with David Braun, now a junior faculty member at Yale New Haven, and he was interested in trying to see if there were differences in immune cell state going from early disease to more advanced disease. And certainly through using single cell transcriptome sequencing and analysis, he was able to find not only worsening T cell exhaustion, but also increased involvement of an M2 of macrophages. So what he identified was in fact a circuit between these two cell types, and using this composite signature look at the prognosis, and was able to find that in fact this presence of this circuit was prognostic for outcome. In another example, we've recently looked in a blood malignancy, so chronic lymphocytic leukemia, again looking at the non-malignant component and trying to use single-cell transcriptome sequencing to evaluate the immune cell state there. And as a comparison, what we did in this study is consider monoclonal B-cell lymphocytosis. So what is considered actually early pre-malignancy, and asking is whether the immune state is already changed even in that pre-malignant state compared to a healthy donor. And we were actually surprised to see that even in MBL, we already see changes in the immune cell composition and particularly in the T cells. So this gives us a sense of the barriers that we have to face as we're trying to give our therapies, our immune modulating therapies to patients with cancer. As we get better and better at identifying T cell clones and TCRs, what do we know about the antigen specificity of those TCRs? Do we understand what the phenotype of those T cell clones are, in addition to their antigen specificity, and how is the T cell immune compartment structured? For this, Giacomo Oliveira in my group recently reported out integrative analysis of CD8 TILs from, these were actually patients that were enrolled on the melanoma neovax trial. So prior to therapy, could we actually characterize the landscape of the CD8 TILs, and specifically trying to focus on those T cells that are tumor specific, because many T cells within the microenvironment may not. So this actually necessitated the generation of patient-derived melanoma cell lines so that he could actually ascertain which TCR-bearing cell was actually reactive to the tumor. And so in doing so, he was able to generate a map of the T cells. He found two broad classes of expression states. So one in the red, orange, and yellow are the exhausted cells. Those in the blue are the non-exhausted memory cells. And what was actually super interesting is that he was able to come up with an approach to actually clone the TCRs en masse, and then developed a combinatorial dye labeling strategy so that we could fastly, through flow cytometry, look at reactivity patterns and do this in a multiplex fashion based on gating of particular cells based on their color. So I'm going through a lot really quickly, but suffice it to say that he actually selected TCRs from this kind of red region versus the blue region. And what he found is that it was the exhausted cells within the tumor that seemed to be 83% mostly tumor specific, and that their reactivities could be against melanoma associated antigens or neoantigens, whereas the blue is really where many of the viral specific T cells were residing in this type of phenotypic compartment. And so really, just to give a visual, these are the melanoma associated reactive, the neoantigen reactive, all in this region up here, whereas the viral reactive are down in this compartment. And so this actually raises the question of, again, what are the T cell, what are the T cell state before you're actually getting into therapy? How can we harness this information to actually design better immunotherapies? The final piece that we're kind of working together in a coordinated fashion is just to think about how can we enhance the immunogenicity of our vaccines. And using this backbone of neovax, we're trying to do a bunch of different add-ons. And so one of the current ongoing trials is adding local IPI, because as this audience knows, it has recruiting and almost enhanced priming functions. And so can we actually do better in terms of priming of a response already? And we're already beginning to start to monitor responses of patients on this ongoing trial, and we're seeing very strong skin responses, and we're looking forward to looking at the profiles of the APCs that, for example, might uptake the vaccine. So now, with all of that activity, has this informed my group in terms of getting back to transplant? So we started thinking about antigens, which got us to thinking about neoantigens, and what about coming back to thinking about minors. And I want to remind you again that when we think about what those donor cells, when they're grafted, what are they seeing? They might be seeing, for example, leukemia cells, but they also may potentially be seeing donor recipient differences on the basis of single nucleotide polymorphisms. And certainly if those SNPs are present on the leukemia cell and are restricted to the blood compartment, then these would be fair game in terms of leukemia antigens. On the other hand, if they're broadly expressed across tissues, as SNPs may, then this would actually set the stage, for example, for maybe a target of graft versus host disease. And so thinking about this, having spent all our time trying to figure out how to create a pipeline for neoantigen prediction, can we not actually also build a pipeline to discover relevant minor histocompatibility antigens? And here we have the added complexity of having another source of DNA in the transplant recipient. So we have the recipient's this part is very similar to a neoantigen pipeline, but now we have the donor. So we have another comparison to make. We also are trying to identify the SNPs by comparison between the recipient and donor. And another important piece is trying to look at expression. So are those SNPs expressed in the leukemia cell as opposed to normal cell? And then can we, for example, subtract out genes that are expressed on non-hematopoietic tissue? And then this can actually get us to the minors. And so we've been hard at work in terms of generating this type of pipeline. We're helped by the plethora of publicly available data that provides many levels of filters so that we can actually get this expression part right. So there are single cell transcriptome data sets across different tissues. There are new proteome data sets, immunopeptidome data sets, and large scale DNA sequencing data. So we have been putting all of that together, and certainly I don't have much data to share with you but certainly we're hard at work of thinking about how this, for example, can help us pinpoint actually common minor histocompatibility antigens to target so that we can enhance graft versus host disease. We're thinking about how a composite score could help us, for example, predict graft versus host disease and apply it to a myriad different research studies so that we can track and monitor what are happening with our patients as they go through their therapeutic interventions. In terms of the immune monitoring, I think you've gathered that's been really part and parcel of what our group has been doing, and we've really been very much supported by the CIMAC-CIDC network. And as many of you know, our group is really focused on trying to get the most we can out of phase 1, phase 2 investigator-initiated immuno-oncology trials that are primarily sponsored by the NCI. And as we started this network, we're in year 5 right now, what we had envisioned was that there would be, for example, trials using different modalities maybe for one particular cancer, and if we could set up our assays in a harmonized fashion, we would have set the stage for a type of comparison that could help us look across these complex assays over many different modalities within a particular cancer type, or think about a modality that is being used across different cancers, and again set the stage for doing that as well. So with that kind of motivation in mind, this is an old figure. There's actually 4 different CIMACs, so Dana-Farber together with MD Anderson, Mount Sinai, and Stanford, and a single data commons. We have been embarking on creating this network, and here's the cast of characters, at least at the Dana-Farber CIMAC. And this is a smattering of some of the clinical trials that we've had the privilege of working on. I'm going to show you some data from 9204, which was a phase 1 study looking at IPI or Nivo for patients after transplant for hemalignancies. Another study that's closely aligned, which is 10026, which is not only adding IPI after transplant, but also adding epigenetic priming, so decitabine. We've also had the privilege of working with solid tumor oncologists, so working with John Schoenfeld on some of his combination radiation transplant trials in colorectal cancer and non-small cell lung cancer, and more recently Annie Silk looking at oncolytic virus. So just to give you a flavor of that, this is the study that we did together with Matt Davids. As I mentioned before, this was looking at patients who had gone through stem cell transplant and upon relapse received immune checkpoint blockade. And because this was a trial of some 50 to 70 patients, of course, across many different diseases, we had a chance to evaluate non-responders, complete responders, transient responders. And what we found was pretty interesting. We were able to access our suite of immune capabilities ranging from flow cytometry to RNA sequencing to mass cytoph and also seromic analysis. And we could look at the T cells themselves, we could look at circulating factors, and we could also look at the microenvironment, and we could identify that in fact ipilimumab, at least in responders, appeared to increase memory T cells, generate subtle changes in the immune repertoire. There was actually a quite striking change in the chemokine signature, but also finding T cell activation with response. Livius in my group was also able to look at, in an informative case study, a patient who received PD-1 blockade following transplant. So if you kind of follow this curve, this was an AML patient who had relapsed, received PD-1 antibody, and had a transient response before then relapsing. So controlled within the same patient, we could look at response and relapse. And you can see when the Nivo was given. We had the fortune of having 6 longitudinal samples from this individual patient, and you can see that with response there was an expansion of CD4s, and looking at also an inflammatory signature that seemed to be present in this patient in advance of response. On the tumor side, Livius was able to find different transcriptional profiles within the AML blast, but it was really there was, with relapse, there was actually a downregulation of PD-L1. So I think this again kind of speaks to kind of this back and forth between the immune response and the tumor cells over time in relationship to therapy. In terms of response, I think, you know, our appetite has been definitely whetted for what we can learn from single cell analysis. And one of the more robust sort of circumstances that we have lots of samples is actually going back to like the golden oldies and this looking again at the DLI response for chronic myelogenous leukemia. We found very attractive because this is an established immunotherapy, highly clinically annotated. From these trials that we had performed within the Dana-Farber, we had this really repository of longitudinally collected marrow samples that we could access over time, and we know that the marrow is a reservoir for T-cell memory. So as you can see here, we had 6 responders, 6 non-responders, really well balanced with samples collected over time. And this was all from this very rich cell source of bone marrow. And together with a group of Dana Pierre, we performed single-cell RNA sequencing as well as ATAC sequencing. And what we found was pretty amazing. So, you know, out of this close to 100,000 marrow-derived T-cell transcriptomes, we could definitely annotate on the basis of T cell state. We can annotate based on which sample, clinical outcome, and also timing. And I think you can see the regional differences already suggesting that there's actually something interesting to dig out there. And I want to remind you that the field has identified lots of different exhaustion states within T cells. Already there's been descriptions of progenitor exhausted versus terminally exhausted that appear to be quite relevant for response to PD1 therapy, because we know that there's blocking of these different cellular programs. What we found actually across our different T cell clusters is that there was actually very nice association of particular T cell clusters with response in the responder. So here are, on this side, are these two meta clusters where we see expansion in responders following DLI, whereas down here we see another meta cluster with decrement in size with response. And looking at their transcriptional profiles, we were able to see profiles that were more consistent with either a progenitor exhausted or terminally exhausted scoring. And so what we were able to see is clear shifts in cell type composition with expansion of the progenitor exhausted populations in the responders. And what was very reassuring is that even in a patient with a different disease, so this is a patient with CLL, not CML for DLI, again, where we had a very prolonged 12-year response to the DLI before ultimate relapse, we can see actually that there is, again, expansion of these progenitor exhausted with response, and then the terminally exhausted coming up with relapse. And so I think next up in terms of where we are going with this is actually beginning to look at other cell types, exploring the spatial organization of cell populations within the marrow, and expanding out to looking at other diseases that are more commonly treated with DLI in the current day and age, namely AML. In the final few moments, I just wanted to kind of talk about how does all this information, how do we kind of put it all together? And I think the two themes that continue to excite me is to think about how we can really continue to move in this track of thinking about personalization of therapy, and how do we think about combinations. And so I think, I think as a field, we've kind of started, at least for vaccines, we've thought about how do we get to shared tumor antigens? Can we not be so personal? Can we use kind of allogeneic cell line, for example, as a source of vaccine? And I think as we think about personalization, one way is to kind of stick on the autologous tumor cells, and certainly there are efforts in that direction. But what I've shown you is this kind of directionality of trying to get to actually the antigenic determinants and going toward personal tumor neoantigens. And so I think this is still, we're continuing to work on this quite in a quite dedicated fashion. I want to point out that this, while we've gone through the paces of an initial proof of concept study, I think we really need to think of better formats of the vaccine and delivery, because in the end of the day, it's the timing and the cost. We're dealing with patients with very advanced disease. We can't have a manufacture time that is taking forever, because that really won't be able to be feasible or make an impact on our patients. And so I think that, again, our world has changed. I think that the fact that we already had ongoing efforts thinking about therapeutic cancer vaccines actually helped mobilize the effort to rapidly generate the vaccines that we've been getting for SARS-CoV-2. So in the example of the Pfizer vaccine, Ugur Sahin had already been working on the RNA platform for cancer, and so I think it was a quick divert in January 2020 to start to work on SARS-CoV-2. I think now we're at a time where maybe we could take the lessons learned from this huge global stressor And maybe this can be brought to bear as we think about this very hard problem of cancer therapeutic vaccine. So how do these various groups have actually, it's been like a global experiment. We have received vaccines of different formats. And so how do we overcome the hurdles of time, cost and time?
Dr. Wu, if you can grab it up.
Yep. I think this is my second to last slide. I think the last part is thinking about combinations. And so this actually has been the inspiration of our group's involvement in the U54 immunoengineering program, which is thinking about how we can use biomaterials, for example, to generate better cellular therapy that can also be put together with vaccines. And finally, I think that going forward in thinking about personalization and combinations, I look forward to the day that we can actually glean information from biopsies that can tell us about the immune profile and state of our patients so that we can better discern what are the actual right combinations to choose for our patients with all our different therapeutic modalities. And so it takes a village. I thank everyone who's been involved in the work, and I am more than happy to take questions.
Thank you very much for this exciting presentation. We have a variety of questions in the Q&A box. If you want to make a question, please place it there, not in the chat. And I'm going to try to filter some of the most representative ones for the sake of time. The first one is from Ingun Stromes from the University of Minnesota. I know that. And she's asking, what is the extent that neoantigen vaccines are inducing T-cell responses? Is there already an induction of some neoantigen-specific T-cells in cancer patients prior to vaccination? Thank you.
That's a great question, Ingrid. Nice to hear from you. We in our peripheral blood monitoring, we actually saw circulating responses already with very little stimulation within the first 3 weeks of vaccine. And I think one interpretation is that there probably is a very, very low frequency response that's already there. But even though when we took the baseline cells and we tried to do stimulation, we couldn't see anything. But I think the fact that there was such a rapid response suggests that there was probably something there that could be expanded. That being said, looking at the TCR repertoire, I think that we are inducing new responses as well. So I think it's a combination.
Thank you. We have another question. What might be the most important negative factor that prevent or suppress the effectiveness of antigen-specific vaccines?
Yeah, so I think a big player, and I kind of got into this a little bit, is kind of trying to understand the immune microenvironment. So I think there's a lot of focus right now on exhausted T cells. I think that increasingly we're cognizant that the myeloids play a role too. I think about some of our patients that we've studied on glioblastoma, where we were happy to see that vaccine was able to push T cells into, the intracranial space. But I think we cannot ignore that we're confronted with this large wall of suppressive myeloid cells. And so what are the immune modulatory modalities that we're going to use to confront that? And I think it's going to be different per disease and the stage of disease that they're in.
I'm going to stick to the mic and ask a question myself, if I might. Do you have evidence of immunoediting? For instance, in patients for which you observe some initial response and then the tumor somehow evolves to lose the antigen? Because that could give us better information about what antigens could be more difficult for.
Absolutely. We're looking, but I would say that we still have sparse data. For, I think that there is some good analysis down the line where if we could kind of amass the experiences of many different groups and try to look at that. But we don't have enough data ourselves to be able to convincingly say that.
So Jose, this is probably the last question.
Fantastic. Thank you very much for this fantastic presentation and tour of force. And we are looking forward to your progress and to learning from your experience and moving vaccines to prevent cancer and recurrences. So thank you.
So thank you, Jose, for your moderation. Thank you, Catherine, for an excellent talk. We will save the questions that were unanswered and provide them to Catherine for her consideration, at least.
Okay.
And
Yeah.
then I would like to remind everyone to please consider attending our next Cancer Moonshot Seminar Series by Dr. Ram, Improving Implementation of Genetic Testing for Lynch Syndrome. And with that, thank you again, Dr. Wu.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.